We've talked a lot about the research. We've talked about all these super interesting findings and the theory that you've developed. But what's all of this without answers and ways of actually helping patients to improve? So you are developing a drug called Mitodicure. It's aiming to be the world's first ME-CFS and post-COVID ME-CFS drug. So please now tell us a bit about it. And, for example, what will be, first of all, its mechanism of action?
MTC-002, our drug, stimulates the sodium pump, the sodium potassium ATPase, and the mitochondrial sodium calcium exchanger, abbreviated NCLX. Stimulation of the first, the sodium pump, prevents the rise in intracellular sodium in skeletal muscle that would cause calcium overload and damage. Stimulation of the mitochondrial NCX directly prevents calcium overload of the mitochondrium. Both effects prevent calcium overload and the associated damage. MTC-002 also raises cerebral blood flow and skeletal muscle perfusion as independent effects.
Well, that sounds really encouraging. So all of these problems in the sodium-potassium pump leading to the sodium overload, which, to spell it out for people, that creates very weak muscles, muscles that don't have a lot of force, that can't endure. And then the calcium overload, which of course is the main mechanism of post-exertional malaise and the muscle tissue death that can occur, which of course is the main reason why you can feel so terrible during PEM. This pill, from what you're saying, will actually prevent those mechanisms. So that is extremely exciting. And by doing so, you would actually be targeting, it seems, the central mechanisms of post-exertional malaise itself. Let's make this more concrete for people, Professor Wirth. Imagine you're an ME-CFS patient. Let's say you're quite a moderate one. Maybe you're able to do 2,500, 3,000 steps a day. You're mainly housebound. You can get PEM quite easily. For example, you just walk up the stairs a bit too quickly. You start taking Mitodicure. What would you imagine or hypothesize would be the changes that that patient would experience?
If the drug is effective and in the way we think, the patient would start with more energy and could perform longer and better. The PEM threshold would get higher in the first time of the treatment. In this phase, the patient should still be careful using the higher energy level that he may feel he has with the drug. The aim is healing the disease. This means that the mitochondrial disturbance has to heal. Mitochondrial damage occurs with every PEM. PEM has to be absolutely avoided, even it would occur at a higher level of effort with the drug. With MTC-002, we would abolish the damaging pathomechanism so that the mitochondrion could recover and regenerate undisturbed. Mitochondria can only regenerate from the pool of intact mitochondria that would be left. Thus, it is important to avoid every effort that causes PEM for a while, even under treatment, to prevent any repeated mitochondrial damage in this recovery phase. Pacing would remain important for healing, at least for a certain time period under treatment. We don't know how long this healing process would take, in which the patient should only carefully raise his activities until healing has hopefully occurred.
The aim is really, as you say, and I want people to really hear this, the aim of Mitodicure is to heal the disease. I mean, so many medications, the aim is more to suppress symptoms, but in this case, you've chosen to target mechanisms of PEM, which really are what keep people stuck in the illness, because every time you go into PEM, all the problems heighten and intensify, and the body then just has this massive repair job that it has to do in order to actually recover from that onslaught. But if you have a pill that can actually make PEM much less likely, well, then the body's still trying to heal, and it will now be able to put those resources into actually healing up the rest of the disease. So do you think it might be feasible that, for example, people could take Mitodicure for a year and then they taper off it and they might actually remain well and free of ME-CFS?
One has to distinguish three terms or mechanisms. There are numerous risk factors for ME-CFS that are inborn or acquired risk factors. Then there are triggers, infection or trauma, for instance, an accident or very stressful situations. These trigger the disease. Triggers in the presence of risk factors cause ME-CFS, which becomes a very stable vicious circle, mainly driven by skeletal muscle mitochondrial dysfunction and perfusion disturbances. The original triggers disappear, but the vicious circle is so stable that it remains because it is self-inducing, self-maintaining. The risk factors also remain. ME-CFS in turn aggravates the risk factors, as already mentioned. An example is POTS, it's a risk factor, but then becomes aggravated by ME-CFS, and POTS further worsens ME-CFS. Our drug is meant to interrupt the vicious circle. As soon as the mitochondrial situation has fully recovered, the patient should be healed. The question is whether ME-CFS could return. Could there be a relapse? The patient has risk factors, and with a strong trigger, there could be relapse indeed. The probability of a relapse would depend on the strength and the number of risk factors, and on the strength of the future triggers in an individual patient, so it's probably an individual matter. My prediction would be that some patients would be healed and could discontinue taking the drugs after a certain treatment period, while in others it would be a lifelong treatment. From a regulatory point of view, we would be compelled to show what happens after discontinuation of the drugs, at least first in the milder or moderate cases.
I've alluded to it just now, but there's so many things going on in ME-CFS. When you were thinking about developing a therapeutic compound for the illness, why did you think that the mechanisms of PEM, that was where you should focus your efforts and that's where you should target the illness with a therapeutic?
Yeah, PEM means calcium overload and damage. Damage may only be mild and thus remain functional depending on the level of exercise. Severe damage, however, can cause morphological damage to mitochondria and even cause necrosis of skeletal muscle cells, as shown in the exercise study, the Dutch exercise study. PEM always means new mitochondrial damage, which may not always be severe, but repeated mild damage could accumulate and become worse and worse to finally cause severe ME-CFS. Healing and improvement require the absence of PEM. In severe cases, the skeletal muscle is in a state of depolarization, which reinforces the pre-existing vicious circle and mitochondrial dysfunction.
We've talked now about how Mitodicure could, well, fingers crossed, we need the scientific studies to show how it works, but, fingers crossed, it could be a game-changing treatment for ME-CFS patients, for long COVID patients, for the millions of people worldwide who are currently suffering so much. It could also help in other ways too, couldn't it? Because what this would be employing would be the so-called pharmacological approach to research, the drug development approach to research, which is something the ME-CFS world hasn't really had up until now. So can you tell us a bit about that, and why might drug development-based research really help advance our understanding of what ME-CFS actually is?
ME-CFS is not a new disease, but a young research field. I've never seen that there is an academic consensus so early on the pathomechanisms, particularly not in such a complex disease. An agent that is derived from a reasonable working hypothesis could finally prove or perhaps disprove the hypothesis. Everybody having a hypothesis is invited to present a therapeutic concept and to develop it for testing in patients. Such competition is what made drug research so effective.
What you're saying is that if there is an hypothesis of an illness, a reasonable working concept of an illness, and you have a therapeutic that's actually based on that hypothesis, and then you test the therapeutic and it works as you expect, not only do you have something to help patients, but you actually have validated the disease model. So you can then say with much more certainty, this is what ME-CFS actually is. And in that way, you kind of speed up research by decades, potentially.
Yeah, you're right.
So tell us, please now, where is Mitodicure at in terms of its current development?
Yeah, this MTC-002 is in the so-called preclinical phase. In this phase, safety, tolerability, and drug metabolism are tested, and a reliable production of the drug is established. Only after this phase can the drug be tested in humans, first in healthy volunteers, and only thereafter in patients in a so-called phase 2A study to test it for efficacy. These investigations before the patient 2A study are all required from a regulatory point of view, and are more or less routine examinations.
And so briefly, Professor Wirth, if you got funding tomorrow, how long are we talking about?
Yeah, it's at least 2.5 years away from a patient study.
One final question for you, Professor Wirth. it's not easy to get funding in the ME-CFS world. There are so many obstacles to getting it, and it's not an easy world to make progress in scientifically. What motivates you to keep going?
I want to see a treatment for this debilitating disease. I mean, I've worked such a long time on it, and I will continue my efforts till I have this drug, and if I know that it works or it doesn't work.
Thank you, Professor Wirth. Well, listen, we all hope for those trials to happen. And fingers crossed that when they do happen, we will see that this drug actually will help the many people around the world who are struggling, some of them very severe, living in darkened rooms, not getting very much support, bedridden. This will be the first medication that could work for people en masse. And so we're all rooting for you, and we're all very grateful for the work that you are doing for the ME-CFS and Long Covid world. And I just want to thank you very much indeed for joining me today.
It was a pleasure for me.